Yeah, it's the AACR Coleman Outstanding Investigator of the Year for Breast Cancer. It's a Lifetime Achievement Award, but for those under 50 years of age. And it really reflects on people who've worked long-term in breast cancer and done translational research that has impacted the patient. It's a great honor to receive this award. There are many, many great candidates out there that I'm aware of, although perhaps not many under 50. This is a business that it takes a while to roll up a large number of good manuscripts, but it's a wonderful, wonderful recognition of the work that I've done over the years. Yes, so in the last few years, there's been a tremendous breakthrough in genetics of breast cancer, and we're talking about the germline. So these are hereditary or inherited genes that influence breast cancer rather than the tumor-type mutations. There are a number of different levels of breast cancer genes impacting different levels of risk. So the main ones that people talk about are BRCA1 and BRCA2, and I was involved actually in the identification of those genes many, many years ago. But since then, there have been maybe 16 to 20 additional genes that have been identified. And one of the issues is that we don't understand how much risk is associated with a mutation in each one. So it's very confusing for the physician and for the patient when a result comes back from a clinical test where you can't interpret the mutation. You really don't know what to tell the patient other than they're at somewhat increased risk of breast cancer. So we've been working with a number of different groups to try and resolve some of those questions, and our biggest partner right now is Ambry Genetics. They're one of the large clinical testing groups in the United States, and they were able to provide us with a testing data set of 121,000 individuals. And from that, we were able to extract 38,000 Caucasian breast cancer patients, female breast cancer patients, which is a huge number, an order of magnitude bigger than the next biggest study that I've seen published. And we're able to use that and then estimate risks for a series of different genes on these genetic testing panels. And there were some surprises. Clearly, BRCA1 and BRCA2 behaved exactly as we expected. Some of the genes that we've known, ATM, CHEK2, and PALB2, were really validated as moderate and high-risk genes, respectively. But we found a few new ones. And these are genes that have been on the panel, so obviously people thought they were involved with breast cancer. But nobody's ever really put a number on that. We don't know how much risk. So in this case, we were able to show that the RAD51D gene and the BARD1 gene are now fairly clearly moderate risk breast cancer genes. And that might reflect a lifetime risk of greater than 20%, and therefore might then impact their risk management through MRI instead of mammograms and steps like that. We also show that the MSH6 gene, which is normally associated with colorectal cancer and endometrial cancer, we actually found that there was an elevated risk for breast cancer for that gene as well. So that's quite interesting because it differs from the other members of that protein complex, and we need more research to figure out what it really means. Furthermore, we excluded a few genes. There were some genes there that everybody just accepted were breast cancer genes. And in particular, I'm talking about the MRN complex, so MRE11, RAD50, and NBN. They've been around for a long time. They were found in a small number of breast cancer families, and they appear to track with breast cancer in the families, the mutations in these genes, and that's why we took them as being breast cancer genes. But we find in this large study that really they have negligible risk effects. They really don't increase the risk of breast cancer at all in this setting. Now more studies are needed there because there are a few unusual situations. There's one particular mutation in the NBN gene that was seen in the Slavic population, and that one is a moderate risk variant, a single variant in the gene that isn't associated with a moderate risk. But yet when we look across the whole gene, it's not. So we have to figure out, is there specific effects or specific mutations, or was there some other complications in the studies that have been done to date? So more studies are needed, even though these numbers seem tremendously high. There are clearly more studies to be done, and I think we're looking for 3, 4, 5 times more patients to really resolve some of those rare genes. Even with 38,000 patients, some of these genes we only had about 10 individuals with mutations. So to really resolve the questions for those individuals, we know we need those bigger numbers to try and get the right answers. Yeah, I think so, and just high throughput testing. And there's a number of companies that are doing this now, and a number of large research projects that are doing this. The clinical testing companies tend to be testing the individuals who qualified for genetic testing, at least in the United States. So these people tend to have a family history of breast cancer or be diagnosed with triple-negative breast cancer. There are also studies ongoing now in the academic area including my own lab, where we're looking at large population-based series. So these are routine women off the street with breast cancer, if you will, and not enriched for family history. And we're trying to look at those and see what the risks in those individuals might be. And that will give us a clear baseline for all these genes in the general population. And then there'll probably be a bump for those that have the family history as well. They'll be a little bit higher than that. We're already there to some extent. There's been numerous trials done with PARP inhibitors and cisplatin in BRCA1 and BRCA2 carriers in both breast and ovarian cancer, mainly in triple negative breast. The other genes we're not so sure about just yet. There's some more studies have been done in ovarian cancer looking at RAD51C, RAD51D, some of those genes that we now think are high-risk ovarian cancer genes but moderate-risk breast cancer genes. And it really does look like they may be responding to these same drugs again. But again, for those particular patients, we're going to need probably new trials or additional trials that will look at large numbers of those mutation carriers, people with mutations in specific genes, and get enough of those patients in a trial to see if there's a specific response. Because it might differ, it might change from gene to gene, even though they're all in the same signaling pathway within the cells. That's a great question. I mean, one of the issues is the privacy of your data. How do you protect your data and make sure that it's used properly? Finding people who can really interpret your data most usefully. And then the type of data you're talking about would potentially be useful for other diseases downstream as well. So I think in the future we'll see much of the population will be getting these types of sequencing, maybe not in the next year or two, but 5 years, 10 years, 20 years from now. Many of us will be walking around with a little stick with a lot of information on it, a lot of sequencing information on it, and it'll tell us about all kinds of different diseases that we're predisposed to, that we have mutations that will drive, things like that. So the future is rich in terms of genetics, that's for certain. But there's a lot of other details that go with it, the privacy that I was talking about, the interpretation of the data. We don't want people misinterpreting the data and having mistakes being made. We really have to do this cautiously and carefully. The conclusion is that I think we've gone a long way to resolving what are the genes that really do drive breast cancer in the population. But I should comment that these are the genes that are known today. And if I take a random sample of breast cancer families right now, only 50% of them are accounted for by these genes. So there's still more breast cancer genes to be found that we have no idea about right now. And there are many academic research studies being done right now to try and find new breast cancer genes that will account for these other 50% of the families. So the story's not over. There's a long way to go yet. We really want to try and account for every family if at all possible, so that relatives can benefit from predictive testing, and hopefully that there'll be therapies going along with some of these things.